Okay, what we're going to do now is go into the questions that we had talked about earlier and answer those questions which have come up over the past 8 years that we've been working together, and for both the practitioners and the patients as well. So let's start with the first question. How well does the Greek test results agree with the therapy recommendations from other genetic testing sites such as that in Seoul, South Korea?
Well, I'm not in the appropriate position to make an assessment for a specific test. It would not be ethical. But in principle, many times tests are agreed, they are aligned to their outcomes, but many times they are not. And pretty much all of us on our section, on our field, we know why. The issue is that when you perform a genetic analysis in the level of DNA or the expression of genes, which is we call epigenetics, we have an outcome which is not always linear with the outcome of the analysis of proteins. We must not forget that all therapies are not targeted, are not designed to block genes or the expression of genes, but actually they are designed to block proteins. So from the analysis we need to be accurate concerning this profile. So when there is a linear relationship between the gene expression or the gene profile and the protein profile, then our test is totally aligned with the results of other laboratories. But when there is no linear relationship, then our test is deviated. And this is a common problem which in principle we must use what we call several platforms. So we need at least 2 platforms, 2 different protocols of analysis in order to have an accurate result. And this is something that we are compliant with this. So whatever we get as an outcome from the microarray analysis, which is one of the basic protocols which detects the gene expression profile of all genome in circulated tumor cells, we verify this outcome in a protein level by expose the cells to the agent that the gene expression profile indicates. And if the outcome is correct, then that's fine. But if the outcome is completely different or irrelevant with the genetic profile, then we need to agree that the viability or cytotoxicity assay is the most accurate one, because this is the outcome that we seek with our test.
Okay, you had mentioned in your explanation about linearity. For the clinicians, could you explain how that could affect the results and everything that we would find as far as the results were concerned?
Yes, of course. This is let's answer with a simple explanation. We know that the DNA is a library, actually has all the information of the structure of the cells and in principle and in general the construction of and function of all organisms. Then we have the expression of this library through molecules that it's the mRNA, and then we have a third step, which is the protein synthesis, and even a fourth step, which is the maturation and the post-translational processes. So the 2 steps, the 2 last steps, are not taken under consideration, which means that we have the genetic library, then we have an expression rate as the first step. So sometimes when the second and third step taken place, they do not express in an analog form, which means that no matter if we have an overexpression of mRNA, you may not have similar overexpression of protein levels. And even though you may not have similar overexpression in functionality of the protein, because even there is a fourth step, which is the glycosylation, the additional groups which they need to be added to the protein so that the protein will become functional. So the last steps are not taken consideration in any genetic test. That's why you need to have a second or even a third platform to confirm it.
Okay, so obviously linearity is very important.
Oh yes,
in
of course,
the final
definitely.
outcome. Yes.